Okay, so welcome back after lunch. So let's begin the afternoon session. Today we'll have the first talk by one of our co-organizers who we're delighted to have here, Mithun, who will be giving, I guess this will be a tutorial?
Yeah, it's pedagogical.
Yeah,
Both my talks are, yeah.
Right. So go ahead.
Thank you, Shakti. Right, so what I thought is that, we've been hearing about, from Shashi yesterday, and also then from Shomdattadi, we heard about, patterns and how patterns form, and how one sort of takes this cell, this single cell, which is a isotropic homogeneous system, and then starts to form patterns and starts to relay positional information, and then how that leads to this cascade of events that leads to patterning and tissue formation and development, essentially, right? So what I thought I'll do is that I'll take this and sort of try to put some, I'll try to frame it, put it inside a mathematical framework, okay, so that you know that when people say that yes, a gradient forms, how does it form precisely? How do I describe the mathematics of that gradient formation? When people say that, I have reactions and I have diffusion, and they somehow interact to form a pattern, what sort of mathematical theories can I construct to sort of explain that pattern formation? Okay, so that's what I'll do. So it's going to be today and tomorrow, essentially. I'll sort of continue. It's going to be one big talk in that sense. And again, this is mostly pedagogical, so please feel free to stop and ask me at any point. I'm happy to stop. It has, unfortunately, a fair bit of maths because I'm putting it inside a mathematical framework. And I know a lot of you are biology undergraduates, but hopefully, I'll start slowly. And again, please stop me and ask if at some point the maths becomes too much, and I'm happy to sort of try to see if I can say it in some other way, okay? So, all right. Thank you. मैं कर देता हूँ. Right. So again, going back to sort of the early days of biological physics, I think somebody, one of yesterday's speakers was also showing this book, What is Life by Erwin Schrödinger, and where he asked this question that how can events in space and time which take place within the boundaries of a living organism be accounted for by physics and chemistry, okay? So this is people, physicists have been in some sense thinking about biological systems for a long time now, including Schrödinger, and this question beautifully encapsulates in some sense the spirit of this workshop in a very simple way. So, what we are talking about, of course, this you can translate this to a full-grown organism as well, but for simplicity, we'll think about a single cell. So that is my boundaries of this living organism. And here I have physical and chemical events taking place. Chemical events as in chemical reactions, as I have chemical reactions, and physical events in that I have motion, okay, of these reactants, of these products. And together, they interact in some way to form structures, to form patterns, okay. So that's the central thing that we'll be looking at today. Before I start, I just want to say that, again, since a lot of you are undergraduates and in some sense beyond developmental biology, you might want to think about how to think about modeling biological systems using tools of physics. I teach a course an elective course to our undergrads, the physics of biological systems, which is up on YouTube, so for any of you who are interested. I also teach a slightly advanced course, which is a stochastic processes approach to biology, which talks about master equations, Fokker-Planck equations, and how to take that machinery and answer biological questions. So that's the second one over there. But this left one is the more sort of, it's a basic course which follows this very nice book by Rob Phillips, Physical Biology of the Cell, and by Nelson, Biological Physics, to sort of take very simple biological systems and think about how to translate that biological, or the questions in biology, how to translate that into a physics language, and then how do I go about solving that. All right. So again, just to reiterate what Shashi was saying yesterday, what the central question that I have over here is that I have this isotropic and homogeneous single cell, which is this oocyte over here, or the fertilized embryo, which develops from this. And what I want to get to is something where I have encoded positional information into the embryo via these gradients. So in this case, in the case of Drosophila, which is what I'll mostly be talking about, this is the gradient of the bicoid protein. Okay. And how do you go from this isotropic system, this homogeneous system, into this system where you encode positional information? How do you break this symmetry and go there? And again, as we discussed yesterday, how you go there is you can have various routes to achieving this. You could self-organize, or you could break this symmetry by certain external information that you encode. For example, in the case of the Drosophila, you have this bicoid mRNA that is deposited at one end, and that breaks the symmetry between the anterior and the posterior, right? So that establishes one anterior-posterior axis. In general, the framework that we will use is that of reactions and diffusion. So that is this chemical reaction plus motion as in diffusion. And again, in the morning, over the two talks in the morning, Mohit and Ramre, we heard a lot about these complex reactions among these gene regulatory networks, feedback loops, and so on that is taking place. So we have these reactants and products being produced through these regulatory networks, and then those things have to move, right? And those things have to move. So there's this motion of proteins, or more generally what Turing called as morphogens, and together these will give rise to patterns. Okay, so that's the basic idea. Now, I thought that again, since this is mostly a pedagogical talk, I'll talk a little bit about diffusion I mean, diffusion is something that all of us know, but it is nice to know perhaps a little bit formally. All right. So, what distinguishes diffusion from directed motion? Anyone? Any of the biology students? What is diffusion? Yes? Very good. So it is passive. Huh?
Along the gradient.
Along the gradient. Along the gradient of what?
Maximum to minimum.
Hmm? Maxima to minima. Okay. So maxima to minima. Anyone else? Correct. That is actually related to this maxima to minima, in that if you have a concentration profile, for example, if you have a concentration profile that looks something like this, maybe, what diffusion is going to try to do is that it is going to try to transport stuff from these regions into these regions, and therefore, after sufficiently long time has passed, you're going to get some sort of a homogeneous uniform concentration. So these two things are linked, but indeed it is a diffusion in that sense is a homogenizing sort of a force. Anyone else? Anything else? Any other characteristics? Can I associate a speed with diffusion when I say that how fast is something moving? Can I associate a speed with diffusion? Yes? So when I say how fast is diffusion, what characterizes that is my diffusion coefficient, right? This quantity d. And what is the units of d? Anyone? Not you, Mark. Anyone? What is diffusion? What is the units of diffusion? Okay, what is the units of velocity? If I'm moving with a speed, what is the units of velocity? Meters per second, right? That tells me how fast I'm moving, 10 meters per second versus 100 meters per second. Does diffusion have the same units as this? Yes, no? No, right? So this has units of length square over time, right? So something like meter square over second, or centimeter square over second, okay? What that tells you is that fundamentally, if I'm moving in a directed fashion with some speed, the physics of that is very different to if I'm diffusing, okay? So I'll try to talk a little bit about diffusion and how. So another way to say that is that, for example, if I were to, I'll say that when I come to that, okay? So I'll talk a little bit about diffusion first before I talk about pattern formation. Yes.
So because diffusion also, it basically transports from stuff, so shouldn't it also have a concentration term, say something related to mass, volume, like concentration stuff in it also?
So there are two aspects over here, and you're absolutely right. I mean, when things are diffusing, it depends on the differences in concentration or differences in mass density or so on. So there are two aspects over there. So what does diffusion do? Diffusion will take a profile like this and try to homogenize that, right? Like you said. So that is one aspect. So that is this gradient of concentration that comes in. The other aspect is that how fast will it do that? So that aspect, that how fast will it do that, is encoded in the diffusion coefficient. Okay, so there are these two aspects, and indeed together is where we'll get the diffusion equation. So this is, in some sense, diffusion is this prototypical statistical mechanics problem, because again, when we do statistical mechanics, what we sort of do is that we take some microscopic particle definition, right? If you think back to, all of you have done some thermodynamics, I hope, ideal gas laws that we did way back in some time, that you have a box of gas and your particles in this box are moving randomly in that sense. You don't want to know the positions and velocities of each particle, but you want to know some macroscopic properties, what is pressure, volume, temperature, and so on and so forth, right? So this prescription, this route that takes you from this microscopic description to this macroscopic, and there's a macroscopic in a probabilistic sense. This macroscopic definition is like at the heart of statistical mechanics, and this diffusion is in some sense a prototypical problem of that, okay? So let me consider this problem, right? So I have a one-dimensional line, and I have a person, a random walker, who can hop to the left or right of this line, right? It can hop to the right with some probability p, and it can hop to the left with some probability q, okay? What does that mean? That means that if I'm looking at trajectories, if I'm, just like I would take Newton's equations of motion and solve that to get a trajectory x as a function of time that would arise from Newton's laws, here the trajectory of my particle, or here the trajectory of my random walker, is defined by this, that wherever it was at the previous time, plus the step that it takes at time t, right? And what is this step at time t? This step at time t, let's say in this model that I have said, is it with probability p, it hops to the right, and with probability q, it hops to the left. Okay? Is that fine? So this is my microscopic trajectories. Okay? It is not a deterministic trajectory, it's a stochastic trajectory because the underlying process is stochastic. Okay? You can hop to the left or right with some probabilities, and therefore this defines my trajectory. But I can move away from this picture and think of a sort of description in terms of these probability densities, that what is the probability that I am at some position x at some time t. Okay? And again, I can write that. So for example, where was I at the previous instant of time? If at the previous instant of time, t minus delta t, I was to the left, then I could come to x. So if I was at x minus delta x, I could come to x with this probability p. Or if I was at right at the same time, t minus delta t, you could hop to the left with the probability q, right? So that's my probabilistic description. You can take the continuum limit of this. Okay, what does that mean? That means you do a Taylor series expansion. Okay, so p of x minus delta x, t minus delta t, right, is p of xt minus delta x del p del x plus delta x whole square by 2 del 2p del x2 minus delta t del p del t, plus higher order terms, of course, plus higher order terms. Okay. Is everybody familiar with the Taylor series? More or less? If no, please, ask. So the idea is that you expand about this point, which is x of t. I have expanded to second order in space, so I've kept terms delta x and terms delta x square, and I've expanded to first order in time, delta t. Okay. So that comes from this term, x minus delta x. Similarly, I'll have something from this term where, and this term has a small p in front of it, small p in front of it, right, small p. And from the other term, I'll have something which has a q times delta x del p del x plus q times delta x square by 2 del 2p del x2 minus q times delta t del p del t. Okay. Just to simplify my life, let me take p equal to q. Okay. So then if p is equal to q, I hop to the left or right with equal probabilities. What happens? This minus p delta x and this plus q delta x, these two terms cancel each other, right? And therefore what I'm left with is this term del 2p del x2. Okay, and I'm left with this term del p del t. So then if I put it back together, if I take this continuum limit with this p equal to q, what I get is this equation, del p del t is something into del 2p del x2. And what is that something? Over here, if you do this correctly, this something is delta x square by delta t. Okay. That is my diffusion, and that is what I call as my diffusion coefficient in the limit that I take small steps and small jumps. Okay. So this is where the scaling of the diffusion coefficient comes. That the diffusion coefficient has the scaling of length square over time, okay, as opposed to length over time. So you take this simple random, this is called a random walk. You take the simple process where you hop to the left or to the right. You take the continuum limit of that, and what you end up with is the diffusion equation. Okay. You could do this p not equal to q, and then what would happen is that you would get an additional velocity term, where this velocity is the difference in how likely are you to move to the left or to the right. Correct? Okay. What does that mean? So now that I have the diffusion equation, I could solve this diffusion equation. And if you solve this diffusion equation, you get this Gaussian, right? E to the power of minus x squared by 4 dt. What does that mean? So what consequences does that have? What consequences that has is that if I calculate the average distance that you have traveled, right? So if I say that how much have I traveled in time t on an average, and again, this is the limit where p and q are same. So I'm equally likely to hop to the left or to the right. Therefore, the average distance that you travel is 0, right? On an average, you haven't gone anywhere. But does that mean that you have not explored space? Of course, you have explored space. And how do you then characterize that? You characterize that by looking at the second moment. What is average of x square? And the average of x square now grows with time. Okay. So as opposed to directed motion, where the average x that you travel grows with time, that is this case, and therefore you can define a velocity, vt. In this case, when you are diffusing, your average of x square grows with time, and the constant over here is diffusion. Okay. So it's a fundamentally different mechanism by which you explore space as opposed to a directed motion. And this thing is sort of universal in the sense that, you know, I started with this hopping approach of whether I go to the left or right. I could put in different microscopic rules. It does not matter. I could choose my step size from some distribution. I could choose when I make that step from some distribution. As long as those distributions obeyed certain properties, I will always end up with something that looks like this, that is diffusive. Okay. So in that sense, what you have is this microscopic behavior, or a variety of microscopic behaviors that gives rise to this sort of universal scaling that x squared grows as time. Okay. All right. Let me not talk about this entropic approach. All right. So what does that mean? So what that means is that if you look at particle trajectories, so here is, for example, a trajectory of a particle. This could be a protein that is diffusing inside this single cell, or it could be an organism itself. And you plot this average of the square, the mean squared displacement. That is why when people talk about diffusion, people talk about this quantity, the mean squared displacement. You will see that for normal diffusion, you will have this thing growing as t to the power of 1. MSD grows as t to the power of 1. Of course, you know, there are always deviations. You could have things that are super diffusive or subdiffusive, depending on whether this exponent is if I call this t to the power of alpha, whether this alpha is greater than 1 or less than 1. So here, something like this is where you have directed motion. So for example, if you're moving with a velocity v, that alpha would be 2, because you would have grown, x squared would have grown as t squared. This is not just, this is of course not just in this context of protein motion, you know, organisms. So for example, this is I think E. coli, this is a classic result from Berg, where again you have the same, scaling of the mean square displacement. So you see this in various contexts in biology, but for today we'll stick to this pattern formation. All right. So with that sort of background on diffusion, you know, it is a homogenizing force. The mean squared, the average displacement is 0, but the mean squared displacement is finite and grows linearly with time. Let us now look at this Bicoid gradient again. And I think, again, you have seen this yesterday as well. So let's take a look at this once again and see that what are we trying to model. Okay. So here is a Drosophila embryo. Here is a Drosophila embryo. What is labeled over here is this protein bicoid. And what you can see is that if you just look at the intensity of light, okay, so the bicoid is fluorescently tagged, and if you look at the intensity of light, what you'll see is that it is highest at this location, which is my anterior pole, where the mRNA is deposited. And then as you go towards the posterior pole, your intensity sort of dies down. The movie is playing in a very spotty fashion, forgive me for that. And what is happening going on over here? So these are the nuclei. So Drosophila is, yes? Sorry, okay. Drosophila is an example of a system which is syncytial in that if you have this embryo over here, you have these nuclei. You have these nuclei which do not have cell walls around them. Okay, and then as these nuclei divide, these are called nuclear cycles. After a certain number of cycles, after about 8 cycles, these nuclei start moving to the surface. Okay, so what you end up with after 9 cycles of division is something like this, where the nuclei have moved to the surface. Okay. Right. These are the nuclei inside the Drosophila cell. So when the embryo is fertilized, you start off with a single nucleus. That nucleus divides, your genetic material gets divided into 2, and you produce 2 nuclei. Then those 2 produce 4, and so on and so forth. So each division is a nuclear cycle. Okay. So this contains the genetic material of Drosophila. Okay. So after about 9 nuclear cycles, this Drosophila, these nuclei come to the surface over here, and then further divisions from 9 to 13 it sort of divides also, it divides on the surface. So, the next nuclear cycle, you get double the number of nuclei, like so. Right. And the next nuclear cycle, you get one more. Yes, Ran.
So there, it seemed like while the movie was playing, there seemed to be some sort of a transcriptional burst near about when
where there's this collective motion of all of these nuclei.
And, like right now, like suddenly bicoid expression sort of spreads out, and then there is a like sort of a burst of bicoid gradient going through. So like so what I was thinking is basically if the transcription is sort of coupled to the cell cycle, and when like this division is happening, bicoid translation rates are lower, then what will happen is the wavefront of the bicoid gradient will also shift over time. And then if that happens, then how do these cells sort of retain what identity they should have?
So it is true that in these initial phases you will see this sort of motion is what you're talking about right here, right? The fact that these nuclei itself seem to be moving in a correlated fashion. It is true that these nuclei move in a correlated fashion, but if you look at the bicoid gradient, and I'll show you the bicoid gradient, how does it evolve with time, you'll see that these temporary fluctuations which happen when this interface to metaphase transition happens, these are very short-lived in that sense. So the overall bicoid gradient is fairly stable and has this nice shape. Okay. So in that sense, yes, there are strong fluctuations, but that is not to disregard that statement. Yes, there are strong fluctuations and correlated motion of all of these nuclei during these development cycles. But it seems that the positional firstly, the bicoid gradient is stable over long times in spite of these fluctuations, and how the positional information is encoded also seems to be fairly robust.
Also, maybe the nuclei being syncytial here helps out in sort of looking at the gradient stably, because like the nucleus, since like the cell membrane not being there, probably they see a uniform gradient throughout.
I mean, definitely the fact that you do not have a cell membrane. So like I was saying, if you had cellularization, this would be in individual compartments, but in this case that is not the case. You have some furrows over here, but they do not enclose. So you have this common cytoplasm, and that is indeed crucial for this bicoid gradient to be formed. The fact that this gradient forms is because whatever bicoid is being produced here from the mRNA at the anterior pole, this is my posterior pole, can diffuse through this common cytoplasm in order to form a gradient. As it diffuses, it also gets trapped inside these nuclei. It enters into these nuclei, which is why you'll see that these nuclei seem to be marked so prominently in the movie, right? So these are very bright spots marked in the movie. That is because this bicoid protein enters inside the nuclear envelope and gets localized there as well. So not only is there a gradient along the anterior to posterior axis, there is also a localization of these bicoid inside the nuclei. Okay. So here is my sort of system. So like I said, I have this common cytoplasm. And what is inside is my yolk, which sort of phase segregates out from the cytoplasm. The bicoid does not, if you will see again this movie, the bicoid does not really enter the bulk of the yolk, right? Mostly all your white spots, all your fluorescent intensities are in this cortical region itself, not inside the yolk. So this diffusion is happening therefore in a cortical region near the surface, not throughout the entire volume of this yolk. Not throughout the entire three-dimensional volume of the yolk. Okay? All right. Right. Now, if you take this experiment and you sort of quantify the gradient of bicoid through these nuclear cycles, so this cycle 9, 10, 11, 12, 13, 14. 14 is where cells form actually. So cellularization happens and you close off these things. And then other downstream genes, Shashi was talking yesterday about downstream genes, hunchback, cripple, and so on, those start getting activated. But the bicoid gradient forms prior to cellularization, and in these times, nuclear cycles 9 to 13, 14, you'll see this is the intensity of the bicoid gradient as a function of position. So this is X is this axis. So So this axis is my x, right? As I go from the anterior to the posterior, this is my x, normalized by the length scale, of the embryo. And you can see that, so this is early time, so this is minutes over here. It's also helpful to sort of know that over what time scales this pattern forms. So this is minutes, so 50 minutes, 100 minutes, 150 minutes. So it's about 2 to 3 hours. These first 13 nuclear cycles takes about 2 to 3 hours, and it is over these time scales that this bicoid gradient is being formed. Okay. It's good to know the time scales because it's also good to have a sort of check as to whether diffusion in this sense is a viable mechanism for forming these patterns, right? How do I go about thinking that? So for example, if you know what are biological diffusion coefficients, biological diffusion coefficients are roughly of the order of micron square per second, okay? Some number, but roughly of the order of micron squares per second. And what that means is that, so let's say, I don't know, let's say 10 micron square per second. Okay. What that means is that if you have something that is happening over the scale of, let's say, 3 hours, okay, then how much distance can it explore? That will be given by this relation, right? X squared is 2 dt. So x square average is 2 dt, so d into t. So if your d is 10 micron square per second and your time is whatever 3 hours, so 3 into 60 into 60. Okay, so that gives you a sense of how much. So how much is this? 18, 36, 36, right? So therefore, if you say that what is the typical size that you can sample through diffusion of this magnitude, where the diffusion coefficient is given by this magnitude and you have available time of this much, then roughly you can scan of the order of hundreds of microns, right, which is the square root of this order of magnitude. Anyway. And therefore you could ask that, well, how large is a Drosophila embryo? And how large is a Drosophila embryo? A Drosophila embryo typically, while Drosophila melanogasters is around 500 microns. Okay? So the length scales are consistent. If the Drosophila embryo was, I don't know, 1 centimeter, and you had to transmit this positional information through diffusion, where the diffusion coefficient had this magnitude, micron square per second, then diffusion would no longer be a sufficient mechanism, because you could not reach a centimeter long object in these time scales through diffusion. But as it turns out, thank you. Could you lose as it turns out, given the length scales and time scales that are involved, diffusion is indeed a mechanism that will allow you to sample the full extent of this Drosophila embryo. Okay. So now, having said all of this, now we come to this gradient formation. How do I then take this? How do I then mathematically write this gradient formation in Drosophila? Okay. So what are the key ingredients that goes in? So one is of course diffusion. So it diffuses along this AP axis. It diffuses in this cortical cytoplasm along this AP axis, and it also typically degrades. So every protein will have some characteristic lifetime, after which it will degrade. So it will have some typical lifetime tau. Okay. So in that case, what I would have is an equation like this, right? So I had written, we had written our diffusion equation, which was del C del T equal to D del 2C del X2, right? That is what, from the random walk, that is what my diffusion equation that we got. So that is of course there, but now I have a degradation that I have a probability that this bicoid will degrade, and it will degrade with some characteristic time scale tau. Okay. Or if you prefer it with some rate, which is the inverse of the time scale, with some rate kappa, let us say. Okay. So this is then my simplest differential equation, simplest mathematical equation that I can write, that will talk about the dynamics of this bicoid protein in this sort of a Drosophila embryo. Now, in order to solve this, you need one additional extra ingredient, and what is that? That is the fact that you have a source of bicoid at the anterior pole, right? So, if you remember the curve that Shashi was showing yesterday, if you plotted the mRNA concentration, it would look something like this. So, it's localized at the pole. To a first order approximation, you can say that, well, it's localized at x equal to 0. I see it's written, so I could write it like this, which essentially says that the concentration is maximum at 0 or at the anterior pole, or you could say that it is being produced at some rate. So I can write the boundary condition as this. Minus d del c del x, that is my flux, is some rate. So it's being produced at some rate q, let us say, at x equal to 0. So at x equal to 0. So both of these are sort of equivalent statements, that my concentration is maximum at the source, because at the source is where this is being produced with some rate. So together, this is called as the synthesis diffusion degradation model. Because you have synthesis over here, bicoid is being produced at the anterior pole. You have diffusion, once produced, it diffuses, and you have degradation, so it has a typical lifetime, tau, and together these three things is going to give rise to a time scale. Right? Now you can solve this equation, of course, fully, but the easier thing to do probably is to look at the steady state. So what does that mean? That means that if I waited long enough, I have this SDD equation, the synthesis diffusion degradation. If I waited long enough, what would the profile look like? What does it mean if I waited long enough? If I waited long enough, that means that things no longer change with time. So that means del C del T is 0. Okay? So this is my steady state. And what does it mean to wait long enough? It means that you have waited a time that is longer than the characteristic time scales in the system. And what is the characteristic time scale in the system? The characteristic time scale in the system over here is, in some sense, the lifetime of the protein. Okay. So if you waited long enough, such that the concentration no longer changes, then of course I have this simple equation, right? That d del 2c del x2 is equal to kappa c. And how does this look? What is the solution of this equation, anyone? Anyone remembers? Exponentials, exactly, right? So you will get C of x is some exponentials. With an exponential means it will have some characteristic length scale, right? It will have some characteristic length scale. And what is that characteristic length scale? That characteristic length scale, so if I take this d down over here, the characteristic length scale is square root d by kappa, okay? Or if I write in terms of the lifetime, d tau. What that means, therefore, this very simple model that I have something which is being produced at one point, that morphogen then diffuses and it degrades, will lead to an exponential profile like this. Will lead to an exponential profile like this, similar to what you see in these experiments, right? And the length scale of this exponential profile is determined by two things: the diffusion coefficient d, and the lifetime tau, Yes. No, of course not. So, for example, again, Shashi was showing these downstream genes, right? Those are not exponential profiles because they are not following this mechanism of this. But bicoid as such, where you have been produced at one point and then you are diffusing. But given the fact that I have a profile like this in the case of bicoid, what is a sort of minimal model that will give you an exponential profile is something like this, where you have synthesis, diffusion, and degradation. You can then, depending on what morphogen specifically you're looking to model, and if it has some other sort of spatial profiles, you can incorporate other physical features that you think are important in order to get the correct sort of description of that particular morphogen profile. Sorry, the steady state solution?
If you want the steady state solution for this equation, then it will always be exponential.
For this equation, of course, it will always be exponential. The question is whether this equation will describe the formation of all morphogen gradients.
Right.
And the answer is no. Right. So here is what I have for the bicoid steady state. It is something into e to the power of minus x by lambda, where this lambda is this. So lambda is the square root d over kappa. Okay. And you can put in sort of the numbers, and I've put in some numbers. These numbers themselves are a source of much controversy because different experiments have given us different numbers. But let us say I put in some average numbers of what comes out from experiments. Again, the diffusion coefficient is of the order of micron square per second. The lifetime is of the order of some 30 minutes or so. So it's around 3,000 seconds. And therefore, if you put this into this expression, square root d tau, what you will get as a length scale for this gradient is around 100 microns. And indeed, that is consistent with what you see in experiments. If you look at Drosophila melanogaster and you characterize the experimental profile, you'll see that indeed characteristic length scales are indeed of the order of 100 microns. Does that mean?
During the transient time period also. Yeah.
I mean, absolutely. So, unclear. So it looks like in real Drosophila, if you look at these later cycles, it seems that
because it's like rising, right?
It's rising,
yeah,
but
so
if you
can it like take the information from?
Absolutely. So in principle, and this is in fact a valid hypothesis, which people call as the pre-steady state hypothesis. That pre-steady state hypothesis. That it's not that I should solve this equation in the steady state, but I should look at the full time dependent solution of that, and maybe this further downstream patterning that happens takes in this positional information not at the steady state, but at some time point where these patterns have not been formed. Sorry, where this length scale has not stabilized. So that is always possible, and you could people have looked at that. It does seem, at least for the case of bicoid, that of course it is rising, but if you look at these final cycles, the changes, so if you look at this orange point and the red point, there is not a lot of change. So it seems that at least even if it is not technically steady state, it is approaching steady state in the case of bicoid.
So the downstream targets are what? They are activated after the
bicoid gradient. Well, steady state is a physics term, let us say, but after some of these initial syncytial cycles.
Okay.
Right. So like I say, so this is an extremely simplistic model. You just put in the minimal ingredients that are required to sort of get what you see in the experiments. Does that mean that it explains sort of everything? The answer is no. There are various issues. I'll just talk about one of them, which relates to scaling. Okay. So for example, here is, and again, these are all taken from this Rob Phillips, this very nice book. So these are patterns, these are intensity profiles from 3 different species. Okay. Bucksky, Melanogaster, sericata. Okay. And the difference is that the embryo length in each of these species is different. So, Drosophila, like I said, is around 500, 600 microns over here. This Sericata over here is much bigger. It is around 1500 microns. Muskie is much smaller. It's around 400 microns, 300, 400 microns or so. What this SDD model says is that I do not have a L dependence in this answer. This C, I do not have a L dependence in this answer. I get a length scale lambda, which depends only on your diffusion coefficient and on your degradation. What that means is that even if I had two systems, one of length L1 and another of length L2, if this were purely the mechanism, in both of these, I would see my length scale, my characteristic length scale to be the same. Okay? But that does not seem to be the case, as you can see from these different profiles. And if you extract sort of characteristic length scales from these three different species, you'll see that there is some sort of a scaling of the characteristic wavelength with the embryo length. Okay? So as the embryo length increases, it seems that the characteristic length scale also increases. You could do this, or rather people have done this in other smart ways as well. For example, you could select for large embryo, so even within a single species. So for example, if you take Drosophila, when I say Drosophila length is roughly 500 microns, it's 500 microns, it's some distribution, right? 500 plus minus something. So what you could do is that you could select for these small embryos, so which are on one end of this distribution. So if you have some distribution of Drosophila embryo lengths, P of L versus L, maybe the mean is around 500 microns, but you have smaller embryos and you have larger embryos, right? So if you carefully select only the small embryos and look at patterns over there, the length scale in those, or if you select only large embryos and then look at the length scale in those, again what you see is that, so this is unscaled, so one embryo is roughly of the length 500, the other is of the length 700. If you now scale them, so if you change your x-axis to x over L, so you scale by the length of the embryo, you see that they fall on one another. What that means is that, again, in this case, what it says is that even within a single species, if you just look at small embryos versus large embryos, what you see is that indeed the characteristic length scale does seem to scale with the length of the embryo. That's what this curve is saying. Okay. So while the SDD model is in some sense a starting point often for when we think about pattern formation in Drosophila or in similar systems, like you said, you have to add different ingredients to it in order to understand the full complexity of the, yes, Rani.
The small embryos don't fall exactly on at the very anterior end. It's just slightly shifted.
Yeah, absolutely, absolutely, and that is because what I have solved is that I have a delta function source in that wherever it is that my source is at x equal to 0.
Yes.
But if you think about it, I have this mRNA that is deposited, and that mRNA is not at x equal to 0. The mRNA is a protein that has some spread. So that there is some spread associated with the mRNA itself as well.
That should be reflected in the larger embryo. If we look at the I figure, the peak for the small is somewhere around, let's say 10 or something. The same X, like mRNA deposition should also matter for the large embryo, but the peak there is much more closer to the anterior.
Absolutely. So that I am not very sure as to the fact that there is this fudging of the source, where the source is, and the source has some
size,
spread, that matters if you were to exactly understand these profiles. But from my experience looking at these profiles for a large number of embryos, close to the source, so whenever we say it's an exponential profile, we sort of leave a region close to the source. Close to the source, there's a lot of fluctuations. But indeed, the size thing will play a role both in the large embryos as well as in the small embryos. There is, I suppose, how much it has spread, or how much the source has. So the source is an mRNA molecule, right? And that itself is diffusing in principle, right? So that itself will have some spread. how much that spread is relative to the size of the embryo is different in these two embryos. So in that sense, there is a distinction even in the source size, the scaled source size, as it were, between these two, between the large and the small embryos as well.
And in the case where you were showing that in Drosophila sericata and other species, so where this size dependence is coming from, so what does one invoke to explain it? Is it some sort of motor dependent transport or cytoplasmic streaming?
So people have tried to invoke various things. I think, so here I have something, alternate mechanisms. But in that sense, it's still not very clear as to what causes this scaling. So in that sense, it's still open. People do have various proposals, but as far as I know, there is no definitive answer to this question, that what causes this scaling. Yes. In my theoretical model, but not in the actual embryo, right? So. Next slide, I'll talk about the source size as well. Okay, ask me again. Right. So nucleoplasmic shuttling, right?
Sir.
Yes.
So if we were to compare the large embryo and the smaller embryo, so the effect of this basically spread that we are seeing in the mRNA, the source, will it be more? Will it be more? Can we compare basically between large and smaller embryo, whether it will be more in one kind of the embryo and less in others?
Okay, so let me talk about, let me talk about that first. mRNA, right? So here is, if you remember what picture Shashi was showing yesterday, here is what the mRNA looks like. Here is what the mRNA gradient looks like, right? So it's not, of course, it's not a delta function. The maternal mRNA diffuses as well, and therefore it has some spread. It is small, of course, compared to the length of the embryo, but still it has some finite spread. Okay. Now, if you say that, well, what characterizes the extent of this embryo, right? This is all this mRNA. Sorry, not embryo. What characterizes the spread of this mRNA? The mRNA is also diffusing, which means that so the amount of spread of the mRNA itself, right? mRNA itself will be determined by the diffusion coefficient and the time that it has had to spread. Again, this is independent. This is not really length dependent. So if this is the only mechanism, then if I ask what is the scaled length, then that will be something like this in some time. And therefore, for a larger length embryo, it will be a smaller source effectively as compared to a smaller embryo. Okay. Sorry, you were asking something about this spread of the mRNA. Size of the source. And so if you think about, so bicoid has this exponential profile, right? So if I think about this, what sort of a profile will the source have? Purely from theoretical arguments. Anyone? This also seems like an exponential. Anyone else? So one thing is that in the bicoid, what I have is a source. What I have is a source, right, at one end. And I should have probably made this clearer. This is a constant source in that you have this mRNA. The mRNA is producing protein constantly. So you have new bicoid molecules being introduced at the anterior end. As far as the mRNA goes, that is not the case, because the mRNA is what was deposited by the mother. So the amount of mRNA is fixed. No new mRNA is being produced inside the embryo, right? So you have this embryo, some mRNA was deposited by the mother, and what that mRNA does is simply now it diffuses, right? So naively, sort of, if I were to make a zeroth order approximation, the concentration of the mRNA profile, the concentration of the mRNA profile, see mRNA, that is going to be something like a Gaussian with some defined spread, right? Because there is no new production happening. Whatever was deposited there just spreads out because of diffusion. This is the homogeneous, half Gaussian. I mean, you know, it's a Gaussian like here as well as here in that sense. Yeah, half Gaussian. If I'm plotting it x0 to L, it's a half Gaussian. Shift towards the Y. See, there is a difference that it is not directed motion. So if you think about the average position, remember when we were doing diffusion, when I'm thinking about the average position stays at 0, if it is purely diffusive. If there is some advection to it, if things are being carried, of course the peak will shift. But otherwise, if you start with a peak at 0, what diffusion will do is that it will simply spread this out. And finally, of course, it will become flat. But the peak does not shift with time. The peak stays where it was.
The source is at the left.
I mean, you have to remember that this is my embryo, right?
Yeah,
so
yeah.
it's a ellipsoid, and one end of this ellipsoid is where my mRNA is.
Yeah.
Now, of course, it is diffusing, which means that, you know, it's going here, it's diffusing all along from this pole of the ellipsoid.
Yeah.
And what I'm considering as x equal to 0 is, of course, so I'm reducing this problem on a 2D, on this surface into an effective one-dimensional problem. In this effective one-dimensional problem, of course, it's not getting stuck because, because underlying it is the fact that it's spreading on this surface, and therefore there is no hard wall over here that is stopping it from, you know, that is a reflective wall and that is stopping it from going on the other side of that.
Okay.
Right. So if you do that, say that this is a sort of Gaussian, of course, starting from a half Gaussian rather, and then you solve this, you will get some. You'll get some modification to this simple exponential profile. You have an exponential component, but then you have other complicated functions that comes up as well, right? So you can take this simple SDD model and you can build layers of complexity on it. For example, here it's a complexity that you have a finite source with some typical size, which is given by sigma, and how that affects the Bicoid gradient. You could think of other things, which was my first slide, which is this nucleoplasmic shuttling. If you remember the movie, the Bicoid you saw was trapped inside the nucleus as well, right? So there was a large aggregations of Bicoid inside the nucleus. If it is inside the nucleus, it cannot diffuse because it is trapped. So there is a version of there is a proposal about trapping of Bicoid, which is called this nucleoplasmic shuttling, which says that essentially if something is in the cytoplasm, it is free and it can move. But if it gets entrapped inside this nucleus, it converts to what is this bound state, and then it cannot move anymore. So you have two species, a free species and a bound species. In one case, the free species can diffuse, but the bound species is trapped. It can revert back because, of course, when there are nuclear division cycles, the nuclear membrane dissolves, and all the Bicoid that was trapped again gets converted back to free. So there's this continuous conversion between free and trapped Bicoid, and you can, those happen with some rates. And again, you can take that into account. This is this Scopus paper. You can take that into account, and that will give you some modification to this simple exponential profile as well. Okay. So depending on how much biology you put in, you start with this minimal model, but then you start putting in more and more biological input to it, and you can get more and more complex models of gradient formation. Okay. And I'll not talk about this, but just to sort of complete the picture, like we said, so once the Bicoid gradient is formed, there are other downstream genes that respond to this Bicoid gradient. For example, here is hunchback, here is giant, here is scruple. They all read the positional information that is encoded in this Bicoid and then modulate their expression in response to this gradient. Okay. How does it go about doing that? So I'll not work this out, but just to give you a very simple sort of, let's concentrate on one, which is the simpler one, which is this hunchback over here, this red. Essentially what it does is that, it's high in the anterior side, and then there is a sharp drop somewhere around the midpoint, and then it is low on the posterior side. Okay. And if you see the hunchback intensity as a function of the bicoid intensity, you see there is this sigmoidal response, the switch-like response. When bicoid is low, hunchback is low. Once bicoid crosses some threshold intensity, hunchback expression sort of switches on, or hunchback intensity sort of goes up, and you get this sort of sigmoidal profile. You can sort of build a model for this. For example, what happens is that, let me do something. Again, so let's say I have my DNA, and somewhere I have my hunchback gene, which starts, okay. Now this hunchback gene, the production of this hunchback gene, this hunchback protein is regulated by bicoid, and what bicoid does is that it comes and binds to regulatory motifs upstream of the gene, okay. And there are multiple binding sites for bicoid. In particular, there are 6 binding sites for bicoid where it can come and bind to regulate the production of hunchback. Okay. So that is what this cartoon sort of says, that the DNA can be in an on state or an off state, depending on how much it is coiled around nucleosomes or not. In the on state, the bicoid protein has sort of 6 binding sites on DNA, so you can have 1 bicoid bound, 3 bicoids bound, all 6 bicoids bound. And depending on how much bicoid is bound, you can regulate the production of this hunchback protein. This happens in a cooperative fashion, and you can work out the statistical mechanics of that. Let me not get into that, but essentially you have something which looks like a Hill function for those of you who have looked at gene regulatory circuits, and you have this 6 which corresponds to the 6 binding sites. So it's a strong, the Hill exponent is in some sense 6, and if you now plug in this bicoid exponential profile, e to the power. So how much bicoid is there in some particular location is of course regulated by this exponential profile, the SDD model. If I just take the simplest SDD model, e to the power of minus x by lambda. And then if you plug this in response to a simple exponential profile of bicoid like this, what you can get is a sigmoidal profile of hunchback like this, where the 2 things that have gone in is that bicoid has this exponential profile, and then there is this cooperative interaction between bicoid and hunchback production, some sort of a sigmoidal response. These 2 together produce a hunchback profile like this. Okay. So this again will of course affect other genes. So because you were asking, so not everything needs to be exponential. Depending on what factors I incorporate in, I can get other sort of profiles. And this in particular is the hunchback. This is a very simple StatMech sort of a problem. So for those of you, physics, if you've worked out partition functions and so on, you can solve this problem and get that expression over there. All right. So this is it for the bicoid sort of gradient formation. Now I'll move to this slide. So this, what we were talking about, this reaction plus diffusion, because SDD model is in some sense the simplest possible example of a reaction diffusion, right? My reaction is simply this degradation, that I had this bicoid protein, that degrades, okay, so it goes to null with some rate, kappa. That is my chemical reaction, and plus I had diffusion of the bicoid. So this is my reaction, this is my diffusion, okay? But one can think about a more generic framework of reaction and diffusion systems, that what are the general conditions for these reaction diffusion systems to form patterns, and that is what Turing did. So that's what I'll move on to next. But let me take a 2-minute break. If you have any questions about the SDD part, the synthesis diffusion degradation part, feel free to ask me now, while I have some more water.
Hi.
Yes.
I have a question.
Yes, please.
So the genes that are activated downstream of bicoid, do they mostly reach threshold, or some of them also reach, let us say, fold change of bicoid?
Some of them also read?
Fold change, you know, temporal fold change, or the slope of the gradient.
Ideally, you should ask a biologist, but as far as I know, it is both. So there are proteins that can read in some sense that, even like Shashi was saying, it's not just the relative concentration that matters or the relative fold change that matters, but the absolute concentration also matters. But in some cases, the fold change, how much you have changed at a particular location, that also matters. So I think it is both, depending on, there are examples to be found of both. Yes. Okay. All right. So then moving to this more generic framework of reaction diffusion, Turing patterns in that sense. I think this is not working anymore. All right. So again, Mohit was talking in the morning that, you know, you have these genes, and they have these these could be activators, these could be repressors. So let's, you know, let's think of the simple system where I have two genes, okay, X and Y, and they have some interactions, self-interactions as well as mutual interactions. These could be activatory, these could be inhibitory. We'll see what conditions are required for the formation of patterns, but in general there is some self-interaction and there is some mutual interaction between these two species. Okay. So this is the chemical reaction part of my setup. Just as a toy system, let's look at this sort of chemical reaction. Okay, and this is just chosen to make the maths very easy. Okay. So this is dx dt. How does the concentration of this X change with time? So what it is saying is that the more of X you have, the more of X you produce, right? Because it comes with a plus sign, and the more of Y you have, the less of X you produce. So it comes with a minus sign. And similarly, the more of X you have, the more of Y you produce, whereas the more of Y you have, the less of Y you produce. So in that sense, what the interaction between X and Y that is encoded in these set of reactions is that X is an activator. It activates itself and it activates Y, whereas Y is an inhibitor. It inhibits itself and it inhibits X. Okay. So this sort of sign is the inhibitory sign, right? And you could ask that, well, what is the steady state of this system, right? And the steady state again is when things are not changing. So you take dx dt to be 0, dy dt to be 0, and therefore the solutions of this is that X is equal to Y is equal to 1, right? So that's my, that's why this is chosen in this way. It's a very simple steady state, X equal to Y equal to 1. Okay. So right now I have not introduced any diffusion as of now. This is a simple chemical reaction that is taking place between these two species at some particular location in space. Okay. So if I think about this Drosophila embryo, sorry, this is a very weird embryo, but you know, in each location you have this chemical reaction between X and Y taking place. Okay. And this gives you a steady state where X, Y is equal to 1, 1. Okay. This is my steady state. Is this, you could ask that whether the steady state is stable or not. And the way we think about stability is that we perturb the system from this, we give a small perturbation to this system. If it is 1, I give it a small kick, 1 plus epsilon, and then I see whether the perturbation grows in time or not. If it grows in time, then the steady state is unstable because you move away from the steady state. If it decays, the steady state is stable, right? So that's what so you can just check that. So you consider deviations from the steady state, in this case 1, 1. So small x is the deviation, small y is the deviation of y. You substitute that in this equation, you get this sort of a matrix equation. Forget about, it's just that, those two equations written in matrix notation. And then you think about whether you know, these perturbations grow with time or not, right? So you look at the eigenvalues of this matrix. If the eigenvalue is positive, that means that this perturbation grows. If the eigenvalue is negative, it means that the perturbation dies down, and therefore the steady state is stable, right? If you calculate the eigenvalues of this matrix, it has both eigenvalues are -1, which means that in this case the steady state is stable, right? If you perturb this steady state, if you give it some small epsilon kick, that kick will die down and it will come back to this 1, 1 steady state. Okay, so it's stable against perturbations. So now what Turing said is that now let me introduce diffusion. And again, this is the sort of counterintuitive thing about this, that generally, like we discussed, what diffusion is a homogenizing force. That if you have hills and valleys, if you have fluctuations in your concentration, what diffusion will try to do is that it will try to smooth away these fluctuations. Here is something where I have started with a homogeneous state, right? Everywhere along in space, this reaction is taking place, and I have a steady state which is 1, 1. And then I'm going to introduce diffusion. Okay? To introduce diffusion, of course, I need a concept of space. So therefore, let me consider in the simplest toy model these two cells, okay? Cell number 1 and cell number 2. Okay? So the reaction is the same in each of these cells. So x1, y1 is the concentration of these two proteins in cell 1. x2, y2 is the concentration of these two proteins in cell 2. The chemical reaction is exactly what I had written down earlier, 5x minus 6y plus 1 and 6x minus 7y plus 1. In both these cells, it's the same chemical reaction that is happening. But now I have coupled these two cells spatially. Of course, this is not a proper diffusion because you need at least 3 cells to have something like a diffusion. But this is a sort of toy diffusion in that what it does is that if the concentration of x is greater in 1, in cell number 1 as compared to cell number 2, then x2 minus x1 would be negative, and therefore the concentration of x in cell 1 would reduce. So whichever cell had the higher concentration of x, that would tend to come down. Whichever cell had the lower concentration of x, that would tend to come up, because these come with a minus sign, x1 minus x2 in this case. Okay. So it sort of mimics this homogenizing effect of diffusion, that wherever you have more of x, it will take x from there and give it to the next cell. Okay. Similarly, where you have more of y, it will take some y molecules from there and put it in the next cell with different diffusion coefficients, dx and dy. Okay. So this is a toy model, the 2-cell model, where I have this chemical reaction, but now I have introduced this diffusion term. And again, you can now look at deviations from this steady state. This state, that's this homogeneous steady state, that it is 1, 1 everywhere in both these cells. Okay. And what you can do is that you can solve this and find the eigenvalue. Let me look at the largest eigenvalue. I have made a choice of the diffusion coefficient, let's say 0.5 in some arbitrary units. And I look at the other diffusion coefficients. So I plot this largest eigenvalue as a function of this second diffusion coefficient dy. And you'll see that as I make this inhibitor diffusion, because remember my y was the inhibitor, it inhibited itself and it inhibited x. So as I make this inhibitor diffusion coefficient large, what happens is that this largest eigenvalue at some point, so in this case when it is greater than 1, the largest eigenvalue becomes positive, right? Which means that I started off with a situation where I had 2 cells, cell number 1 and cell number 2. In both these, my xi, I had this state, 1, 1 and 1, 1. As long as these were not coupled, this is the steady state of my chemical kinetics. Now I have spatially coupled these 2 cells. Okay, there is crosstalk, there is diffusion between these 2 cells. And there are regions, so but in particular where the inhibitor diffuses faster than this activator, where you can destabilize this homogeneous steady state. And that is when you will get patterns. Okay. So let me see. So let me not think about this formal thing. Maybe I'll show it tomorrow. But let me just show you how that looks like. Okay. So this is before I say. So here is a system which is called Schnackenberg kinetics, which is one of the sort of earliest toy models in the field of pattern formation, where you have some chemical kinetics, and the chemical kinetics is given by that, right? So essentially, I have 2 species, U and V. I have 2 species, U and V. dt and dv dt. And they have this chemical kinetics. Du dt is f of uv and dv dt is g of uv. Okay. And this chemical kinetics is encoded in these equations. So what do these equations say? What this equation says is that there is a constant term, which means that this chemical u is being produced at some rate. This constant, so let's say u, right? So it is being produced at some rate, okay, which is C1. Once it is produced, it can degrade with a rate C minus 1. So it can degrade with a rate C minus 1, right? That is this term, C minus 1 u. Okay. And then there is a reaction which is that u square v, which means that 2u plus v goes to 3u. Okay. So 2 molecules of u, 1 molecule of v comes together to produce 3 molecules of u. So that is the reaction. So that is what this chemical kinetics is saying, that you have this u square v term over here. It has a plus sign for u because you're producing an extra u, and it has a minus sign for v because you're taking away 1 molecule of v, right? And then there is some production of v itself. So there's some constant production of v itself. So that is the chemical kinetics that you have. Okay. So I do v with some C2. Okay. So this plus this plus this. So this set of chemical reactions is what is encoded in these equations. In the absence of any diffusive coupling, so these have a steady state. You can work it out. These have a nice steady state. So therefore, when these cells are not coupled to one another, you have a homogeneous steady state that is present. Now you add diffusion, and contrary to this, like we were saying, contrary to this usual effect of diffusion, what diffusion does in this case is that it destabilizes this homogeneous steady state, and what you'll get is the formation of patterns. Right? So these are the concentrations of u and v as a function of x. Right? So you started off with a homogeneous steady state. As time evolves, so I should have put these on loop. As time evolves, these small fluctuations, so the steady state is no longer stable against fluctuations, and you get these patterns that form. Okay. Which one is this? This is also that. It's just the same system. It's a one-dimensional pattern formation system, x, but now I'm plotting it as a function of time. So initially it was homogeneous. As time evolves, you get these regions of high u and regions of high v that form spontaneously because of this diffusion-driven instabilities. So in this Turing's mechanism, diffusion is not a homogenizing force, but rather what it does in this specific parameter regime where inhibitors diffuse faster than activators, and we can work out the maths a little more formally tomorrow. But generically, the idea is that for Turing pattern formation, diffusion is not a homogenizing force, but it's a destabilizing force. Similarly, you can solve this same set of equations in 2 dimensions. I think not this, sorry. Yeah, so this is a 2-dimensional system. So you have u's and v's are functions of x and y. Initially it was a homogeneous state. As you let time evolve, what you get are these very nice spot-like patterns that form spontaneously. Again, simply because you have coupled these different positions or these different cells through diffusion. Okay. So the simple homogeneous steady state is no longer stable to perturbations, and you get the formation of these spontaneous patterns like this. You can try this out. So for example, if any of you are interested, I have a simple MATLAB code that you can take and you can run, and you can play with the parameters, and you can try to see that, in which regions do you get pattern formation, in which regions do you not get pattern formation. So I can give you that code at some point. Okay. So I think I'll stop here today and then continue on tomorrow.
So, any questions? Okay.
Yes.
like to ask for concerning the first part of the talk. You showed that this bicoid gradient scales with tissue size or
embryo size.
It's kind of old observation, but
yes.
like when you introduce diffusion, there is no scaling there as such. Saying like,
correct.
You just get some lambda.
Correct.
So my question is, because there are different mechanisms of this morphogen spreading, do you know currently what's the how would you explain this scaling in Drosophila? What mechanism is responsible for that?
So so that's what as far as I know, there is no definitive answer to that. That why there is what sort of mechanisms would give rise to this length scaling. I think to a certain extent one has to take the fact that this diffusion is not happening in this homogeneous environment, the fact that there's this heterogeneous background where you have these nuclei, you have these furrows, and therefore you have constrictions, so you have this diffusion is happening against this heterogeneous background. I think that needs to be built in, and how that changes with cell cycle will probably play a role in how this scaling comes about. But again, there is, at least as far as I know, there is no definitive answer to what gives rise to this scaling with the organism size.
Yeah,
yes.
Thanks.
Yes.
I don't know if it's correct or not,
yes.
but talking about scales, can we visualize it as if I have a fixed box of a given size and I have standing waves and their interference gives me peaks or crests and that gives me patterns. So by looking at the size of my box varying along, that would be my length scale, and this will surely differ in the patterns I get. So can we like draw parallels between the two this way?
So then we can talk about it, but in that sense one has to think about how you get the standing waves, right, in this sort of a setup. And that you need to generate standing waves in order to get interference among them, and then therefore generate some sort of a scaling. But that one has to then, you have to come up with some mechanism for generating such waves in the first place.
Thank you, sir.
Yes. Numbers as in numbers of?
Number of molecules, like, so I'm just interested in like how much stochasticity is like involved over here. So is it too stochastic? I mean, like, can it change the that profile, that exponential profile that is known?
So I don't have a number offhand, I can look it up, but the concentrations are high enough that stochastic fluctuations, at least in these later stages, will not change the fundamental nature of the gradient. So it is robust to those. So if you have a few molecules, of course, stochastic fluctuations can play a large role, but this is not in that regime. Okay. So the gradient is fairly robust in that sense. The exact numbers I can look up and check.
And also the volume also like plays some part, right? I mean,
when you can sort of work out, because the embryo length is some 500 microns. It's an ellipsoid. The other side is, I suppose, some 300 microns or so, and it diffuses along the cortex. So the cortical height is roughly some 50-60 microns. So it diffuses in that sort of a shell. So that's the volume that is available to the bicoid to diffuse.
But is it larger enough to kind of use these equations to kind of justify that?
Absolutely, absolutely. So if you so when you say it is large enough or not, you have to compare the volume or the length scales that are involved with the sizes of these proteins, right? So, and in that sense, indeed the volume is large enough that you can think in terms of these continuum approaches, where you talk of concentrations and you talk of diffusion and so on. So that mechanism goes through. There is no issue with that.
Okay, thank you.
Okay, so if there are no further questions, then I guess we'll break for tea now, and we will return here around 4. Okay. And let's thank the speaker once again.